General microbiology, morphology and physiology of bacteria. Eukaryotes are different in causing infection because highly structured cell with organized cell organelles. Prokaryote is bacteria, mycoplasma, blue-green algae. What is eukaryote? Fungus is eukaryote and protozoa is also eukaryote. Prokaryotes are characterized by absence of nuclear membrane. True about bacteria, mitochondria is always absent in bacteria and it is divided by binary fission. Which of the following is a bacteria taxonomically? Chlamydia, rickettsia and mycoplasma are bacteria taxonomically. All the following pathogenic bacteria fulfill Koch postulate except Treponema pallidum. So Treponema pallidum does not follow fulfill the Koch postulate. Other one is Mycobacterium lepra. These don't fulfill Koch postulate. To see the bacteria, methods used are microscopy and stained preparations. Microscope used in microbiology is light microscope, phase contrast microscope and fluorescent microscope. Light microscopy resolution is 300 nanometer. Dark ground microscopy is used to see flagella. Dark ground microscopy is used for detection of spirochetes. Dark ground microscopy is used for TPI. Dye used in fluorescent microscopy is auramine. And one of the following staining method is an example of negative staining, Indian ink preparation. In negative staining, the structure is to be demonstrated is not stained. Wet Indian ink preparation is used for demonstration of capsule, slender organism and spirochetes. Safety pin appearance is shown by Haemophilus ducreyi and Donovani granulomatous. Correct order of gram staining is gentian violet, iodine, then carbonfusion. Which is not a component of gram stain? Methylene blue is not a content of gram stain. The contents are ethanol, iodine and gentian violet. The difference between gram positive and gram negative organism is that gram positive organism contains teichoic acid. Which of the following are acid fast bacilli positive with 5% sulfuric acid? Mycobacterium leprae. Mycobacterium leprae is positive with 5% sulfuric acid. Mycobacterium tuberculosis with 20% and Nocardia with 1%. Acid fast organisms are spores, Nocardia, Legionella and Rhodococcus. Louis Pasteur is not associated with discovery of Mycobacterium tuberculosis. He is associated with the introduction of complex media, discovery of rabies vaccine and disproved spontaneous generation theory. Wet India ink preparation is used to demonstrate capsule. Electron microscopy is used for following except TPI. So electron microscopy is used to differentiate T and B lymphocyte, immunoglobulin G deposit in kidney and flagella. Peptidoglycans are present in gram negative bacteria and gram positive bacteria. Bacterial cell wall is composed of all except teichoic acid. So bacterial cell wall is composed of muramic acid, glucosamine and mucopeptide. Mesosome in bacteria are functional unit for respiratory enzymes. Capsulated organism is Klebsiella and Cryptococcus. So which are the capsulated organisms? Pneumococcus and Pseudomonas, Anthracis bacillus, Klebsiella species, Influenza, Haemophilus, Yersinia species, Bordetella species, Meningococci, Clostridium perfringens, Cryptococcus, Neoformans and Clostridium wilchi and Vibrio parahemolyticus. So you can remember it by uh, Paki YBMCV. Stain. Which stain is not taken by capsule in gram negative organism? Because stain is not taken by capsule in gram negative organism because the capsule consists of polysaccharide and protein. Metachromatic granules are found in diphtheria and Gardnerella vaginalis. Metachromatic granules are stained by Ponder's stain. Bacteria with tuft of flagella at one end are called Lophotricate. Non-motile organism is Shigella dysentery. Microorganism adhere to host cell by lipoic acid, lecithin, lectin, fimbriae, capsule, viral capsid. Organ of adhesion of bacteria is fimbria. Adhesion is useful in bacterial attachment. Flagella, not true, is attachment. Flagella is for locomotion. It is protein in nature and it is antigenic. Peptidoglycans are present in gram-negative and gram-positive bacteria. The cytoplasmic membrane of bacteria is responsible for selective permeability. True about bacterial capsule is following except it stains by gram stain. So what is true about bacterial capsule? It prevents phagocytosis, protects bacteria from lytic enzymes and lost by repeated subcultures. Which are capsulated bacteria? Neisseria, Haemophilus and Streptococcus salivarius. And which is not capsulated? Corynebacterium diphtheria. All are capsulated bacteria except Vibrio cholerae. So which are capsulated? Streptococcus pneumoniae, Klebsiella pneumonia and Haemophilus influenza. The endotoxin which leads to endotoxic shock is actually lipopolysaccharide. Endotoxin from gram-negative organism is lipopolysaccharide. Exotoxins are highly antigenic. True about exotoxin, can be toxoided. Streptolysin O functionally and structurally related to tetanolysin, pneumolysin, Clostridium perfringens toxin, Listeriolysin. Lipopolysaccharide structure is characteristic of endotoxin. Exaltation is increased virulence. Obligatory anaerobes cannot withstand oxygen because of absence superoxide dismutase, catalase and peroxidase. Hot cold phenomenon is seen due to which toxin? Beta lysine. Following grows in cell free medium except Bartonella. So which organisms grow in the cell free medium? Rickettsia, Mycobacterium lepra and Syphilis. The term viable not cultivable is used for Mycobacterium lepra, Treponema pallidum. All of the following organisms are known to survive intracellularly except Streptococcus pyogenes. So which organisms can survive intracellularly? Neisseria meningitidis, Salmonella typhi and Legionella pneumophilia. Which of the following are intracellular? Virus, chlamydia and rickettsia. Mycoplasma is not intracellular. Obligatory intracellular organism is chlamydia. Which one of the following bacteria is classified as facultative anaerobe? Escherichia. The following pathogen does not satisfy Koch's postulate. So Treponema pallidum and Mycobacterium lepra. So lepra bacilli is the answer. Correct sequence of bacterial growth curve is lag phase, then log phase, then stationary phase, then decline phase. Or it can be lag phase, then exponential phase, then log phase, then death phase. Sporulation occurs in stationary phase. Mesophilic organisms are those that grow best in the temperature of 25 degrees Celsius to 40 degrees Celsius. Which one of the following bacteria is classified as facultative anaerobe? Escherichia. Which one of the following is microaerophilic? Campylobacter. Cell wall deficient organism are Mycoplasma. Cell wall structure is found in all except Mycoplasma pneumoniae. So cell wall structure is found in Staphylococcus aureus, Pseudomonas and Corynebacterium, but it is not found in Mycoplasma pneumoniae. Now sterilization and disinfection. Disinfectant kills the following pathogenic microorganisms. Sterilizing agents include dry heat and ethylene oxide. Agent used for sterilization is ethylene oxide, ionizing radiation, 2% glutaraldehyde and formaldehyde with low pressure steam. An antiseptic means an agent applied on skin to eradicate pathogenic microbes. The best skin disinfectant is betadine. Which one of the following can be reliably used for hand washing? Chlorhexidine and isopropyl alcohol. Sterilization of culture media containing serum is by micropore filter. Best method of skin disinfectant is tincture iodine. Phenol coefficient indicates efficacy of a disinfectant. Riedel-Walker coefficient is related with disinfecting power. Heating and subsequent plating is a method used for isolating Clostridia. All are methods of sterilization by dry heat except autoclaving, uh, which are the methods of dry heat: flaming, incineration, and hot air oven. Autoclaving is not a dry heat method, it is a moist heat method. Which of the following is true about pasteurization? It kills all bacteria except thermoduric bacteria. Moist heat kills all of the following except Coxiella burnetii. So moist heat can kill Brucella, Mycobacteria and Salmonella, but it cannot kill Coxiella burnetii. Tyndallization is a type of intermittent sterilization. The best method of sterilization of dusting powder is hot air oven. Bacterial spores are best destroyed by autoclaving at 121 degrees Celsius for 20 minutes. Autoclaving is done steam at 121 degrees Celsius for 15 minutes. Out of the following, the true statement regarding sterilization is dry heat is the best method of sterilization of liquid paraffin. Glass vessels and syringes are best sterilized by hot air oven and autoclaving. Hot air oven is used for sterilization of all except in hot air oven for holding period of 1 hour, temperature required is none of these. Vaccines are sterilized by heat inactivation. Hospital dressing is best disposed of by incineration. Tyndallization is a method of sterilization. 40% formalin is used to sterilize all microbes and spores. A chest physician performs bronchoscopy in the procedure room of the outpatient department to make the instrument safe for use in the next patient waiting outside. The most appropriate method to disinfect the endoscope is by 2% glutaraldehyde for 20 minutes. Endoscope tube is sterilized by glutaraldehyde. The operating temperature in an ethylene oxide sterilization during a warm cycle is 49 to 63 degrees Celsius. What is false about alcohol in disinfection? It has sporicidal activity is false. What is true? Ethanol is used, isopropyl alcohol is used, and it has bactericidal activity. Which of the following is an important disinfectant on account of effectively destroying gram-positive and gram-negative bacteria, viruses, and even spores at low pH level? Chlorine. Heat labile instruments for use in surgical procedures can be best sterilized by ethylene oxide gas. The sterilization method for the instruments which are damaged by dry heat is radiation.